A common epidemic in our society affecting over a third of the population as declared by the National Heart Foundation in 2012. Correct fat storage is impeccable for maintaining health and altering disease states in humans. There is a growing need to determine how we can alter fat storage using pharmacological agents. The nematode worm, Cenobambius elegans, is an excellent organism for testing as they have good balance for drugs that modulate metabolism. This poses the question, can nematodes reduce fat content in humans? Studies conducted by Chow and authors had discovered that a hormone nuclear receptor gene, NHR49, functions as a key regulator of fat consumption. 2 alkaloids were used and both showed that they were effective. This suggests that they predominantly affect lipid synthesis rather than fatty acid B oxidation. In studies conducted by George Lieumeux and colleagues, it was determined that a fat-reducing compound, F17, is dependent on kinase AMPK encoded by the AAK1 gene in Cenobambius elegans. Lieumeux and colleagues exposed wild-type worms to 3,200 compounds for screening. C. elegans were used due to the fact that they are eukaryotic and have transparent properties. 2 days post the screen, worms were exposed to Nile red staining. This staining was used to mark intracellular fat droplets and then observed under the microscope. Several molecules were tested without any obvious effects on feeding, growth or reproduction. Figure 1. This shows the increasing and decreasing compounds under Nile red staining. Here we see F17. Figure 2. This shows how the fat phenotype induced by F17 requires an AMP complex containing the catalytic subunit encoded by AAK1. Figure 3. This figure indicates that F17 and F21 modulate fat metabolism through distinct mechanisms. It is clearly evident that F17 has been successful. Further assessments were to determine whether F17 affected the AMPK pathways in human liver cells. Here, F17 activates the AMPK pathways and the number of liquid droplets in the HEPG2 hepatocarcinoma cells. They are decreased. Therefore, it is identified that Nile red staining and identifying that compound F17 dependent on kinase AMPK encoded by the AAK1 gene in C. elegans and human HEPG2 cells is successful at decreasing storage in both C. elegans and human liver cells. This paves the way for the determination into obesity pharmacological drugs in the future.